All right, so let's move on to nuclear transport. All right, so I asked you all to do some pre-class reading and homework before today. So I hope you all are very prepared to dive in. So the topic that we're trying to address is very similar, parallel to what we've been doing before with delivery, the targeting of proteins to the endoplasmic reticulum or different organelles along the secretory pathway. So we want, in this case, to consider the special case of the nucleus. How do we send proteins to the nucleus or out of the nucleus back to the cytoplasm? All right? So that's the consideration, the problem that we're going to have to deal with. And and there's similarities and differences with the endoplasmic reticulum. So I wanted to stress some of those. Most important from the structural point of view, I want to point out that the nucleus, you can think of the nucleus as an extension of the endoplasmic reticulum. Just think that you have your ball of DNA and you coat it in endoplasmic reticulum. The result of that is that the nucleus is going to have not one membrane like the ER, Golgi, lysosomes, but it's going to be coated by two membranes, two bilayers. So because of that, we could say that the nucleus has a nuclear envelope with an inner membrane and an outer membrane. And you can see that they are completely continuous with the membranes of the endoplasmic reticulum. So it is as if you're just simply extending membranes from the ER to form the nuclear envelope. And therefore, the space that is in between those membranes in the nuclear envelope is continuous fluid that goes into the lumen of the ER. All right? So just think of that as one single topological space. All right? So in considering traffic to the proteins to the nucleus, we're going to have to deal with a totally different beast. Instead of going through one little transmembrane protein across the ER membrane, the translocon, we're going to have to somehow go across those two membranes. All right? It's a very different situation, and therefore the cell has come up with a very different way of dealing with it. So let's go back to Instapol, and I want to ask you where you are in your knowledge of nucleus given the homework. Did you all vote again? All right, so we have a battleground between A and E. All right, so let's go with E. The protein transport into the nucleus occurs across the outer and inner nuclear membranes. I set you up to choose that by what I just said in the previous slide, and that is not a right answer. All right, why not though? I just told you that we have to go across two membranes. Why is that answer wrong? Yes?
Because they actually have to go through the nuclear pore, which is where like the outer and inner membrane kind of fuse and create like basically a hole.
So they actually, to go in through, this is the amazing thing. To go across the nucleus, you don't have to go across any membrane. Why? Because what I omitted from saying in this slide, to set you up, was that the outer membrane and the inner membrane fuse with each other and create actual holes, big gapping holes across which then the transport occurs. And we know that there's zero traffic across the actual bilayers. All right? So all traffic in and out of the nucleus is going to occur through those holes, which we call nuclear pores. So that leaves us with the possibility that a large protein of 100 kilodaltons requires a signal to go from one compartment to the other, which is the theme that we are going through the course, right? That proteins in general, to go from one compartment to another, they're going to have a signal to travel to that compartment. So that is a true statement A. All right?
And similarly, those holes are so big that B, C, and D, small proteins can go across, ions can go across, hydrogen ions can go across, so it's like gateways.
Yeah. So So those holes, however, are not just empty holes of just the fusion of the membranes. You actually have to stabilize them, and we have a gigantic structure called the nuclear pore complex, made up of many, many different types of proteins that form this nuclear pore complex. Because they are in the nuclear pore, many of those proteins are called nucleoporins. And we have more to say about nucleoporins in a minute. But these guys are so large, essentially you can think of it as a gate that you can fit a ribosomal subunit across. That's important because the ribosomal subunits are assembled inside the nucleus with big RNA and RNA proteins that bind to the large RNA subunit or the small RNA subunit, and then those subunits have to then be transported to the cytoplasm where they're going to function in translation. So it has to be big enough to be able to fit ribosomal subunits across. Right? And we have in the audience Dr. Arlen Johnson, who works here at UT, who actually works on ribosomal assembly and transport in and out of the nucleus. So if you have super detailed molecular questions about how that works, there's your person to ask. All right? So here's a conundrum though. Okay, so we have this gigantic hole. We have a hole so big that it allows for big things to go across. Yet, this hole is able to reject the transport of specific things that are much smaller than a ribosome. All right? So how do you build a gate that can differentiate what can go across and what shouldn't go across? All right? So that's the goal for today, to try to understand how that works. And then we're going to continue making it more detailed at next class after the exam. So, what many classic experiments established a long time ago is that the gate of the nuclear pore complex allows for the complete free diffusion of small molecules like ions. And that's why, as Dr. Halloran was pointing out, since ions can move readily across simply by diffusion, totally unimpeded across the nuclear pores, you cannot have any kind of gradient. Right? So proton ions will diffuse, calcium ions will diffuse, ATP, ADP will diffuse. Anything, even small proteins, well, not even small, somewhere under, the cutoff is somewhere on the range of 50 kilodaltons, which is a pretty decent sized protein, right? So if you have a small protein, like some of those small G proteins that Dr. Halloran was talking about, 25 kilodaltons diffuse right across, all right? But if you have something large, say 100 kilodaltons, then it cannot diffuse across. So to move across, then it's going to have a signal. Again, the conundrum is, how do you build a gate that will be able to say, okay, you have a gate, you move across, you're large, but then this other thing that is just as large cannot move across that gate, right? In the early days, some people were suggesting a system kind of like like in spaceships, right, where you have two gates. You open one gate, you allow it to go in the middle, close that gate, and then open the inside doors to move across, right? Turned out to be a lot easier than that. So, to go in, we need a signal. So, just like we have, if you want to go into the ER, you have to have an ERSS. If you want to go to stay in the endoplasmic reticulum once you have been in there, you have to have a kilo. You want to go to the lysosome, send a lysosomal enzyme to the lysosome, you need a pre-test question. Mannose-6-phosphate signal. If you want to go into a clathrin-coated pit by endocytosis, you have to have a tyrosine signal, all right? So to go into the nucleus, we have to have a signal. There's a variety of signals that have been discovered. The only one we're going to consider is called the classic nuclear localization signal, or basic NLS, which is called basic not because it's fundamental, but because it is basic amino acids or positively charged amino acids. So it consists of 5 positively charged amino acids. So they can be lysines or arginines most of the time. And you could have all lysines or all arginines or any combination thereof. The important thing is that these 5 charges have to be exposed on the surface of the protein so that, true of all signals, the signals have to be exposed so that then we can have another protein that can bind to that and acts as a receptor. Right? So we have a signal and we have to have a receptor. So here's an old experiment looking at a viral protein that is followed with, probably with GFP. And in the wild type form, this viral protein goes into the nucleus because it has a nuclear localization signal. So here, what you see is a bunch of nuclei of many cells. You don't see their cytoplasm in there. So just brightly lit nuclei. But if you change even just one of those 5 positively charged amino acids, the protein cannot go into the nucleus. Now the nuclei are empty, and the protein stays in the cytoplasm. All right? So what can we say about those 5 amino acids? Those 5 amino acids are indispensable, so they are going to be necessary. All right? And that's all you need. You don't need anything else beyond those 5 amino acids, 5 positively charged amino acids. So they are sufficient. They're necessary and sufficient. Just like KDEL. You need all those 4 amino acids. You don't need anything else. Necessary and sufficient. So, if we have a signal, again, somebody has to read the signal, and that's going to be another protein. And that protein is we're going to call a receptor. Right? So we had a chiral signal, we had a chiral receptor. Mannose-6-phosphate signal, mannose-6-phosphate receptor. So here we have an NLS. We have to have a NLS receptor. We're going to call it a nuclear import receptor. And since I told you that there's actually a variety of nuclear localization signals, signals that will send proteins into the nucleus, we have to have correspondingly different receptors that recognize each of those different signals. Again, just for simplicity, we're just focusing on the classic 5 positively charged amino acid signal, and there's going to be a receptor protein that binds to that specific signal. What would be your prediction of what amino acids are in the surface of the receptor to bind to the NLS? Aspartate or glutamic acid, negatively charged amino acids, right? And that's indeed the case. All right. So we have a signal and we have a receptor. Notice that these 2 are totally soluble proteins, all right? So we're not dealing with transmembrane segments, membrane binding sites. We are going to have to go totally in the water environment of this, from the cytoplasm to the nucleus, all right? And not going across an actual lipid bilayer, all right? So how do we send these proteins across into the nucleus?